Hi, my name is Eduardo Ceballos, and my company, xBiologix, is a synthetic biology spin-out from Oregon State University, where we've made great strides in the early detection of infectious disease. The COVID pandemic forces complicated and often uncomfortable conversations, especially related to employees returning to work. In the following slides, I'll make the case for a better approach, a flag raising for a new technology whose time, I believe, has come. The pandemic has exposed major shortcomings in the role diagnostics play with respect to controlling health crises. Diagnostics guide treatments, but they are not answering the important question for employees and employers, which is, can I go back to work today? Am I infectious? As a case in point, the asymptomatic are not sick, but they are infectious. This slide shows why it's so challenging. COVID shows up in samples at just 100 virus. Symptoms show days later, and typically a diagnostic happens very late. The employee's experience washes somewhere in the middle of all this. But as you can see, if the test doesn't detect 100 virus, it doesn't tell the employee they're infectious soon enough. So what's an employee to do? The next slide shows us what their options are. Here are all the available tests, all on a level playing field, apples to apples. From the previous slide, we know that we need to detect 100 virus. And if that weren't dismaying enough, we need to do it at very, very often, 25 to 50 million times a day. We know this because from early on, many epidemiologists have been helping us understand just how daunting controlling the spread of COVID is. The Rockefeller Foundation, for example, details that in order to enable contact tracing, the US alone needs up to 50 million tests a day. And this assumes that the tests are being done at home. Okay, so which tests do that? PCR tests are sensitive enough, but they only scale to 1 million tests a day. That's 49 million too few. In other words, to give you an idea, if testing to reboot the entire economy were a marathon, it would be an ultramarathon, a 24-hour race. But in that race, you'd wash out after the first 30 minutes. But PCR deserves a lot of attention here. These ultrasensitive, synthetic biology-fueled molecular tests responded amazingly quickly to the crisis. They showed up as soon as genetic code was available. But again, they don't scale and they're too expensive. Along the bottom of the graph, you see, those are the antigen tests. They do scale well, but they haven't caught up. Plus, you lose a lot of sensitivity. So what we're looking for is up there in the right corner. That's where you're going to find the test you need. A test that repeats 50 million times a day and detects 100 virus. So if you're trying to reboot the economy, if you're trying to get to work, the question to ask yourself is, can the test I take tell me if I'm infectious? So clearly for 49 million people, the answer is no, but there is hope. Suppose it's possible to combine the sensitivity and agility of the synthetic biology that made PCR tests so responsive to the pandemic with the scalability of the antigen test. xBiologix has done just that. We've developed an easy-to-use, in-home antigen test platform that detects diseases before symptoms start in a form factor that guarantees reliability and scalability. Our technology packages together synthetic biology that pushes way past the antigen test, their limits of detection, and reduces the time to market. We combine that with an inexpensive inkjet printed lab on a chip. This makes it possible to diagnose COVID-19 in home before symptoms start and at a scale that matters. To deliver massively scalable antigen tests that are as sensitive as PCR, we outperform conventional strategies. Our platform is 1 million times more sensitive than standard tests. And because we're printing diagnostics, think the microchip revolution. It can multiplex. So imagine testing for flu A, flu B, and COVID in triplicate, in saliva, or in breath. But the key is the agility of our synthetic biology means that we can meet crises just as quickly as PCR did. Under the hood, our labeling and materials technologies dramatically improve the performance and amplify signal in three significant ways. First, our synthetic biology creates orderly surface geometries on a material and breaks through the nanogram barrier to sub-picogram detection. That's a 1,000-fold improvement. Another 1,000-fold comes from choosing the biochemical amplification strategy that yields another improvement. And finally, the physical device itself improves surface to volume ratios to such an extent that it increases the sensitivity another 1,000-fold. These are all additive. Our go-to market strategy leverages the network we're building to develop a B2B beachhead by identifying regional businesses with high capital infrastructures and employees that can't telecommute. So think life sciences or high-tech facilities, hospitals, this sort of thing. We expect to enter the market in mid-2021, leveraging a parallel IP and regulation strategy that optimizes for these aggressive schedules. And there are unusual market dynamics. The FDA has cleared our path with emergency use authorizations, and federal funding to support strategies like ours is really unprecedented. Abbott has set the price for a point-of-care device at about $5. According to the Rockefeller Foundation, COVID will begin to circulate seasonally and require effective contact tracing through 2025. So we believe a market is well established, but as of today, 910 separate efforts are undergone. About half of those are immunoassays, and a fraction of those are tests for antigen. This mostly attests to the need for many, many tests, but like the Abbotts, none of them are ultrasensitive. Our team evolved over years of collaboration with Oregon State University. It combines serial entrepreneurs and an experienced biotech-savvy science team and the thought leaders in synthetic protein synthesis and advanced materials. In our timeline, we've gone way past our go-to-market threshold. We passed it last quarter, and we're planning to launch trials in the near future. In closing, recall where our talk began. We want to join the conversation between the employer and the employee and emphasize how best to align with both their priorities. We want each employee to know their health status, and we want to empower policies that preserve healthy workplaces. Today, we're looking to anchor a life science network right here in Oregon. But to position our fledgling effort, we need to build and navigate an even bigger network. So here we are, executing. Join us. We're ready. Thank you.